One of the very important consequences of interactions between objects in nature is that interactions very often give rise into new properties. One of the things that we will discover over the course is that very often our understanding of the elements that form a system is not enough to understand the system itself. There are properties that cannot be reduced to the properties of the individual units. That's the idea of complexity, and this lecture is about the first approach that we can take in terms of the dynamics to see what happens when things interact and why these interactions often give rise to unexpected results. So interactions, let me give you, take this opportunity to talk about a very important concept, which is complexity. What is complexity? Okay? Although today we'll explore the idea of how pairs of objects, which could be two different kinds of molecules, for example, interact. This is only a first step into a more general context. I've chosen as an example to illustrate the idea of what is complexity, termite mounds, right? Termites are a class of social insects. Social insects are one of the most successful innovations in evolution. They have really conquered the earth in many ways and provide an illustration of what complexity is, right? Let's think about what a termite nest means. Termite nests can be huge structures, of course, there's a range of sizes, but can actually reach the size of the order of meters high. 3 meters, for example, is typical for some species. And who designs that? The structure inside is quite sophisticated in terms of the ventilation system, how the chambers are organized, etc., etc. And clearly, this size, the magnitude of the system, goes far beyond the size of the individuals that build the nests, right? Termites, inside these complex tunnels, have a size of the order of 10 to the minus 3. So it's several orders of magnitude of difference. How do the termites build the nest, right? What do they know about that? Well, termites are blind. They communicate with their nearest nest mates inside the nest, and it's chemical communication. Not a single termite has any idea of how to build the nest. So where is the nest coming from? It comes from what we call collective intelligence. It is the interaction between the agents, and we will see at some point how that might happen when we go into what's called Turing patterns. It's the interactions between the individuals what creates new phenomena, higher scale order, that provides the source for building the structures, okay? And this is a two-direction interaction. Termites construct structures, and as structures get built, the shape of these structures affects the behavior of the termites. So you can see here something really, really important. On the one hand, we cannot reduce the large-scale structure to the understanding of single individuals. On the other hand, is a causal loop here, right? Termites affect the structure, and the structure affect termites, right? A very, very important principle for most complex systems. So, now I'm going to explain you what's the logic of building models where essentially you have two classes of objects that interact in an extremely simple way, right? We are far from cognition or far from building structures so far, right? But the idea that we'll use as a starting point is, imagine I have two kinds of objects, and we will define that in terms of a simple reaction, okay, that interact in some way. The arrow here can capture many different processes, right, and gives some kind of product. Okay, it's a kind of a simple chemical reaction of some kind. And that happens with some efficiency, right? Sometimes we'll use a probability, sometimes it's going to be just a rate. It doesn't matter here right now. And Specifically, we make a number of assumptions here. And at some point, we'll see how some of these assumptions might be too strong and why some of the assumptions, when you relax them, give rise into new phenomena. In my case, let's assume that I have some kind of a container where my A and B objects move around randomly, right? I put this here, this stick, kind of suggesting that this is a well-mixed system. And this is a term that's used very often. We say well-mixed when we want to say we don't have to worry about space, for example. That I'm located here, you are located here, and it's important, right? Imagine that it's just a mixture, right? So I don't have to worry about space. And what I want to know is, if I have this kind of reaction, what is the dynamics of this system? How A and B are going to change over time, right? As they interact and become something else, okay? In this case, if you think about it, it's kind of a decay process, because essentially what I'm seeing here is that A and B combine and disappear into something else, right? It's not the simple model we just define where one object becomes another kind of object. It's more there. All right, I'm going to give you the intuition of how this has to be mapped into a differential equation, okay? All right, thinking in space sometimes helps, because in the end allows us to think in the microscopic mechanisms. For example, A and B need to be close enough to react in some way, okay? So essentially what I want to say is, if I want to calculate these, let's write down this in terms of concentrations, okay? Clearly the change of A over time, given this reaction, has to be negative, okay? Because every time interaction happens, I lose A molecules, right? And in some way, here inside, I had to have A, B, mu, okay? Let me write this way. My parameter, which measures how efficient is the process, and on the other hand, somehow the amounts of A and B, okay? I might use A or A concentration. I'm referring to the same thing, right? It's a complete abuse of language, but that's fine. Okay, one clear thing is, if I think in the original system, right? Imagine here I'm in a state where this is the number of A molecules I have, okay? This is the number of B molecules I have, and let's assume that at this stage, N, this is the total number of A and B molecules I have. This is a changing variable, okay? But let's label this way. So, and I want to make this simple calculation. What is the likelihood that A and B get together, right? So that they can react. If I'm thinking in this scenario here, which is the well-mixed system, not the spatial one, the well-mixed, this is essentially a random process, right? You just move around, and it's exactly the same as if I ask the following question. If this is an urn, right, and this is marbles, and I just sample at random and choose 2 marbles, what is the likelihood that they will be A and B? Because that's what I need for them to interact, okay? So if I want to calculate that, the probability of choosing A is just this number. The probability of choosing B is just this number, just probabilities, okay? And the thing is, what's the probability that simultaneously I see or I choose A and B, right? For the well-mixed scenario, the probability of interaction should be proportional to PA, PB, all right? The probability of finding A and independently finding B, so independent processes, something that I might not be able to assume in this case, right? But essentially what I'm saying is, if you think in PA, PB as the the actual relative concentrations, in fact, I'm just saying that this is essentially the product between one concentration and the other, right? It's going to be proportional to that. And you can see that this makes all sense, because actually what I'm saying is simply that the change in A over time is going to be minus, because it's a decay process, with an efficiency of the reaction that tells me how fast this is a decay process, and then just the products of concentrations. This, in a way, reflects this underlying microscopic logic, and on the other hand, makes total sense. If A or B are not present, this will be zero. There will be no reactions. I need both, right? And the relative concentrations are relevant too, because if B is very large and A is very small, this in a way weights how likely it is for this pair of finding each other in any case, all right? I will have a completely equivalent expression for B, because in the end A and B go into the same dynamics, okay? So that's the simple way of introducing the idea of how dynamically pairwise interactions, right? This is a pairwise interaction, okay, which are going to dominate quite a lot our following lectures. How to define interactions. That's going to be applied for epidemic spreading, for many other processes, right? Okay, let me give you an example, right? Our first example of how to apply that. And I would like to illustrate that with an example that is very interesting for our understanding of genome organization, and has to do with very simple experiments involving hybridization of DNA. The logic of the problem will be the same, but you will see how powerful it is to use a very simple model to approach something that in principle appears very complicated, okay? The idea is the following. Imagine that, and it's a classic experiment, you'll see why this is so interesting. You have a long chain of DNA, okay, we know that the double helix, when you hit you hit a double strand, it becomes separated, right? And that the process, which has a lot of big consequences for things like the PCR reaction, when you reduce the temperature, right, it's kind of an inverse dynamics. So the things that have complementarity find each other. Again, remind that finding each other is important. Okay, you go into the double strand DNA again. This will be double strand DNA, single strand DNA. All right. What I want to do is to define a very simple mathematical approach to something related to these dynamics. Imagine that we do the following. Imagine we take a genome, right, the DNA, we break down it into many pieces, okay? And I want to understand when I use as a starting point, my system at high temperature, so my strands have been broken and separated into single strands, and I reduce the temperature and this gets hybridized, right, into double strand. In other words, imagine I have broken the whole genome into small parts, right? That would be my double strand DNA fragment, okay? I increase temperature, right? So this gets separated into single strands, right? And now, from this state, right, we might time 0. Now I reduce temperature again, so that allow the system to hybridizing. You find your complementary chain. You get into again into the double strand. Okay, and I want to define the dynamics for this. So in other words, I want to make a model of this part. All right? Very well. Well, look at the problem the way it is defined. I start with a whole genome, okay? Could be a single copy of the, for example, the whole genome of E. coli. I break it, oops. So I have actually a set of fragments, which necessarily will be in different sizes. I have not a single size. I just plot here. Each will be a nucleotide, but it could be a range of sizes here. And of course, every chain carries a different sequence. So in principle, I have a huge variability here. A whole set of higher dimensional system, higher dimensional dynamics, because I have multitude of possibilities. So I'm going to ignore that. Right? I'm going to ignore that the fragments have variable size, they have a distribution. I'm going to ignore completely that I have a sequence, and so different energies of actual binding. And I'm going to assume that I can simplify this into something that is just identical chains with the same size. I consider them equivalent, all right? And that they will hybridize with a rate that I'm going to assign this mu here, right? Exactly in the same way. Seems an oversimplification, because it is an oversimplification, but it works very well, right? How do how I build the model here? Well, when hybridization happens, you go from this state to this state in a process that involves how likely is that every strand here finds its complementary, right? It has to be a complementary. We don't assume any degradation. So for every single strand, someone somewhere has to actually attach to that, okay? So what is the dynamic? Suppose that I want to write an equation. Let's call this concentration here. Let me call it S for single strand, okay? The concentration of single strand chains, right? When I reduce the temperature, I'm going to observe these single strands to be going down in time as they find out their pairs, right? So I want to calculate this. The equation for the decay in time of the single strand DNA. So what I'm going to find here is the following. First of all, clearly, this is going to be a process which has a negative rate. So I'm going to reduce over time the presence of every single, of single strand DNA. On the other hand, this is nothing but a reaction, okay? What essentially I'm saying is that single strand plus single strand go into double strand. That's the essential, the toy model I'm playing with. You could say, well, the reality is what you're saying is that a single strand plus, if you want, we can write this here, the complementary, right? Is the complementary strand that I need to attach to, right? Right, so in a way, you could say this is, I'm going to make a trick here. Yeah? Don't take this as a serious mathematical thing. In a way, okay, this is not well defined. In a way, what I'm saying is the process of hybridization requires finding for every single strand the alternative one, right? The complementary. But in fact, what I'm saying is that they need to find out someone there, assuming this completely homogeneous situation, right, which has exactly the same concentration. Think about it. You have a probability of having this particular strand, and the probability of finding the complementary is exactly the same. So actually, the right equation for this, I don't need to make this distinction between strands and kind of the set of complementary strands. I don't have to. Because by definition, by construction, okay, the concentration for each one is exactly the same. And I'm not making any other assumption. I'm not distinguishing one from the other. So that's the equation in this simple toy model that describes the way in which DNA hybridization happens, right? This equation is easy to deal with. It can be separated easily, right, as usual. We start from an initial condition where every single element is single strand, okay? If I call that S0, what I'm essentially going to have is this. Let me write down the solution. Suppose that I put this here. S, or concentration of S at time 0 is going to be S0. Everyone is going to be here, right? Initial condition, okay? If you solve this equation just by integrating, what you'll find is that the solution is this. All right? This is an equation that is different from things that we have seen so far. This is a solution for my differential equation with this particular initial condition. We always need a particular initial condition to solve it, right? You can check easily that this has two things that are necessary to be consistent. For time 0, right, you just have S is S0. And when time proceeds, what you have in the limit is that essentially every single strand is going to vanish, will disappear. This tends to 0, okay? So for time going to infinity, S goes to 0. Okay. So how does it look like in terms of the dynamics, right? And why is this important? What kind of things can reveal? Well, let me show you. Imagine you take E. coli, right? That's a classic experiment. And you do this experiment. Sorry, you do the modeling. Here you have your initial condition, right? And I'm going to use logarithmic coordinates for the x-axis, right? Just a reminder, logarithmic scaling allows actually to expand things so we can see better early events in particular. And that makes more clear how it works. Essentially, this goes slowly here, then decays rapidly, and then goes to 0. Not that quick, but anyway. That would be the shape, right? For this is logarithm time, okay? Let me write it to make it clear. Okay, so that's one thing that is interesting in itself. One definition that you might find in the literature, again, remember, for these dynamical systems, often it's nice to have a single number that in a way captures timescales. One thing you can find in some places is what is the time scale, right, required for achieving the half of the original concentration, okay? It's not important for us right now. Okay, that could be the dynamics, right, for this pairwise interaction when you have decay. One very important discovery that was made at some point is that when you do that, not for bacteria, for single-cell organisms, but for a multicellular system, for example, for mice, you take mice or human DNA, right? And you actually do the same, not the same calculation, the same experiment, right? You take your genome, break it into pieces, and do the hybridization, right? what happens is, that would be original time, this logarithm of time. What was observed in these systems was that actually, when you look at this, you don't have one decay, one single curve here. You have 1 and 2 and 3. That was long before all our golden age of sequencing even was possible, right? What is this revealing? It's revealing something really interesting about genome architecture, okay? Think about the logic that we have been using. We define a dynamics that involves how easy it is for pairwise interactions to happen. Something happened very quickly here, right? It was a decay of 30% maybe of DNA content that was quickly finding the complementary chains, right? Why is this happening? Because this is the small sequences that are highly repeated, right? It's very easy to find your pairwise interactions because it's a lot of them and they are short, right? This is the highly repetitive, repetitive DNA, very short sequences. This is about 10 to the 4th, 10 to the 6th copies. Okay? Here is it's moderately repeated DNA sequences. Okay? This is another part of the genome. This is about objects that have 10 to the 2 copies. Right? And then it's a part of the genome that has a more hard time to get hybridized. Why? Because you have single copies. There are genes that only have single copies. So the parts of the sequence of these genes have to find their partners, which is only a single copy. It's going to take a while to find them, right? Despite this is a probabilistic process, it's a very broad description of the system. It's interesting that thanks for this dynamical description, we know what's the meaning of that. We can say this is the dynamics of hybridization. That's what's happening, right? Clusters of elements that find each other. And on the other hand, it reveals a complex structure for the genome. For the first time, it was possible to actually see that genome architecture, right? These and other evidence, but this in particular reveals that there's an organization that was unsuspected. So even at the level of description of a dynamical system, we can say relevant things about the architecture of genomes. Nothing more, nothing else. We just have seen how to make a very simple model of pairwise interactions where two objects get together and somehow they become a different kind of object, right? For example, single-strand DNA molecules find each other by complementarity and lead to a double-strand DNA. Okay, remember when we analyzed the problem of growth and decay, right? Essentially, we had two different but similar models, right? One was the growth model, and the other one also linear, almost identical, except for the sign, which makes a big difference. One grows to infinity, and the other goes to 0. There's an equivalence between our pairwise interaction models that follows up from this idea. We just seen that. Let me write down now this with x variables without any other kind of distinction of A, B, or whatever. We've seen this model here, right, that we just have solved, and we have seen that this decays in some particular shape that in the logarithm time scale more or less behave like this. Okay? So this allowed us to actually see something when looking at complex genomes. What would be the equivalent here for not decay but growth process? And what would be the context? The context is important for several reasons. There are several case studies where the growth process involving pairwise interactions leads to phenomena that have big relevance in society, in evolution, and in other contexts. Okay? And let me give you as an example the problem of demographic growth, right? The growth of human populations. We humans have been expanding as a successful species in the planet at a very, very accelerated way. So actually, if you follow in time almost any kind of recorded time series, time would be for years, that would be human population for countries or globally, what you will find out is that from industrial revolution times, etc., and before, you have a curve that essentially is quite flat. It was a really slow growth process. And then very steep acceleration, right? Okay, when you look at this, you could think, well, isn't this essentially what I see here, right, for the exponential growth process? Let's remember that if you solve this equation, what you have is an exponential growth, which is really fast, right? But the interesting thing is that when you look at human growth, this is not what we see here, right? If you look at exponential growth of populations of bacteria, that's what fits well the process, right? Essentially, single-cell organisms self-replicating. We don't see that here. This is a different pattern, right? It's much more steep. So what's the origin of that? How do we make a model? And more important, what are the consequences? Okay. One important thing that distinguishes this process from this is that in this case, we have two kinds of individuals, right? A single-cell organism essentially can self-replicate, okay? It doesn't need anyone else to do it. But we have here sexual reproduction. So we essentially have two different kinds, if I use this kind of notation, two different kinds of individuals, right? They need to get together, right? To do what? Well, they may make this in a simple representation as a reaction. They remain there, right? But then I can add new individuals, which can be either one type or the other, right? Man or woman. All right, that is something that happens at some rate. Again, I'm going to use an oversimplified picture of the real thing. The rate of growth of populations depends on a number of externalities, right? Has been changing over time as we have been overcoming different selection barriers. Medicine has advanced, and in general, health has been improving, right? And right now, unfortunately, it still happens in many places in the world, right? But anyway, let's assume that this is essentially a homogeneous process, okay? How do we make a model of that? We already have some clues. We are going to assume, for simplicity, that the system is well mixed, okay? I'm not worrying about the fact that things happen in terms of humans happen with a geography, okay? And for a number of reasons, since the world has been globalizing and geography is less and less important, this approximation in many cases is not bad. So, how do I make a model of this? Again, A and B have to match, right? And if I want to do a model, for me, A is going to label the class of object, but also the number, right? I'm going to use that to make things simple. What's the number of A individuals? That could be man, woman, okay? Well, there are two things happening here. In these events, this is a growth process, so it's not a minus here, it's a plus. And it happens with some efficiency, right? There's a likelihood that interaction happens, okay? And also, this parameter also weights how many offspring I'm generating, right? It averages a number of things, okay? We are not doing a microscopic analysis. I need both A and B, okay? So I need the product. I need them to find each other. Now interactions translate into a multiplication. Both are needed, and their relative interaction depends on their relative concentrations, okay? Well, to be fair, that would be essentially the basic equation. I could write down another equation for B, and of course I need a parameter, an extra parameter or an extra number here, which actually says that once this happens, this interaction leads to either A or B, half of the time, okay? To make things simpler, because we are not very worried about that, let me write down this in this way. Okay. K is a constant of proportionality that captures both the fact that I can generate one of the others, of the different classes, and the efficiency. That's my equation, right? Again, I could say, well, I have two variables here, but now let's go back into another thing that simplifies the model. Essentially, the amount of male and female individuals in a population is basically the same, right? There's a slight difference, but irrelevant here. So if A is very close to B, my model of growth, of demographic growth, is this, which again, as you can see, is exactly the same model that I just wrote down for the hybridization of DNA, right? Pairwise interactions, things have to be found each other, and because of this symmetry, I can simplify the model this way. Except that this is growth, so in that respect, it's completely different. Again, this equation is easy to solve, right? There's no big problem with that. You can do it as an exercise, and what you find out is something really interesting, right? Let's assume that initially my original population, okay, let's say that this is A of 0, was A0. In my notation, right, I would be, the solution would be like this. 1 minus, put K, right? It's basically the same mathematically, except for a sign, which makes sense. But see how dramatic is the difference because of this sign, right? Let's think about it. For time 0, I'm going back into checking out that things have been done properly. For time 0, the number of individuals is the number of individuals at the beginning. No problem. What happens when time grows? Something strange happens here. Because if time grows, clearly there's some number here that is creating something strange, like populations go to negative values. The answer to this strange paradox is the following. Actually, if you analyze this is time, this is the number of individuals. Have in mind that I did that for A, could be for B, so this is going, the total population will be twice, if you want, okay? That will be the number of total individuals, all right? In reality, I start from some initial condition, okay? It's easy to show, and you can do it analytically. You can analyze this system and see that initially the dynamics is quite flat, right? Essentially, it's kind of, I maintain myself very close to A0 for a long time. So it's a delay introduced by this, very characteristic. Then at some point start to rise. But the interesting thing is that actually if you do the mathematical analysis is, as I get close to this, right? So if time grows, I'm going close to this is going to 0, which means that the that the quotient here goes to infinity. In other words, this model, as simple as it looks, contains, I'm going to write down as a TC, contains a so-called singularity, meaning that this grows, and mathematically, this predicts that the system is going to go to infinity. Okay? So, I have a singularity. It doesn't make any sense to think beyond this time because my population has been growing and growing. That's the relevant part of the story. So what are the consequences? What are the limitations? Okay. Well, one interesting thing that we can observe. The dynamics and the presence of a singularity depends on the initial condition, which is kind of a tricky thing. TC, right, which is obtained when we make this 0, all right, interestingly depends on the initial state, which is something that usually we don't see. Remember when we analyzed the time for doubling or the time for halving for these two models, it didn't depend on initial conditions at all. It only depends on parameters. Here, A0 is relevant, okay? Is this realistic? Well, you could say, but it's not, because there's no such a thing as an infinite population, right? In the real world, we have resources involved that limit the growth. And it's true. And you will see in another lecture what happens when you make human populations growing, interacting with resources that are finite. You see that one of the outcomes is collapse, is social and economic collapse. Well, it's not, but again, remember when we analyze the exponential decay or exponential growth, we say these are very rough approximations, but they can give you a lot of information about how actually the system works. All right? So the same happens here. If we have pairwise interactions like this as part of the dynamics, we have to predict that whatever is increasing in a hyper-exponential way. This is what is called either hyperbolic or hyper-exponential. Hyper-exponential growth, meaning that they go faster than exponential, which is a lot because exponential is quite fast, okay? And reveals that there's a finite time where the system achieves an infinite size, right? Something quite unexpected and quite counterintuitive. Well, this will appear in other contexts. We will see at some point that when we model the dynamics of cooperation in complex systems, cooperation is an extremely important part that makes biological complexity in evolution to emerge, right? We'll see that hyperbolic growth is a very important part of the story. You know, to overcome limitations in complexity, these kind of phenomena are important. And I have to mention that because nowadays there's this discussion about the singularity in a context that is not disconnected from that, okay? You can think in terms of these pairwise connections or interactions, right, in the following way. This could be technologies, right? It's important for us, for this bioengineering perspective. If you think in technologies as inventions that can combine to give rise to other inventions, right? That's something that happens in technology. So you could have other objects that combine it, give rise to a new technology, etc., right? This is actually something that is explosive, right? Because you keep combining pairs of things and this goes really into novel technologies that accelerate very quickly. One of the leading scholars that proposed this idea was economist Brian Arthur. He's actually the strongest advocate of this idea that evolution of technology happens by combination. And in the last decade, this kind of picture has been used in the context of thinking in how technology in the future will accelerate, in particular technology for biological systems, right? There's been the extrapolation that since, for example, computers and computer memory has been rapidly increasing in time, in some years in the future we'll have computers that will have as many parts as a human brain. And that has been extrapolated in saying that in a way we are going to singularity. At some point technology will be able to solve everything you can imagine, right? We'll keep discussing about that, right? There's a lot of things involved in that claim that are completely incorrect. But nevertheless, it's interesting to see that in a way, yes, technology is accelerating. And in the context of biology, which is not just computer hardware, we'll see to what extent this idea can be taken as correct.